Vancouver. His lab employs population genomic approaches and field and greenhouse experiments to study the origin and evolution of plant species, exploit the genetic diversity of wild extremophile species for crop improvement, and combat invasive weeds, focusing on wild and domesticated sunflowers. Rieseberg is a climate analytic, highly cited researcher, and his work has been recognized by MacArthur and Guggenheim Fellowships and the Darwin Wallace Medal in Evolutionary Biology. He is an elected fellow on the Royal Societies of London and Canada, the Norwegian Academy of Arts and Letters, and the US National Academy of Science. He is a past president of the American Genetic Association and the Botanical Society of America, and has served as chief editor of Molecular Ecology. Professor Rieseberg, please, you can proceed. Maybe about half an hour if possible. Yes.
Thanks for that very kind introduction, and I also want to thank particularly Professor Zhao Jun for organizing, and the organizing committee for organizing this incredible conference and inviting me to speak. I'm so pleased to be here in China. I've had a wonderful time so far, seeing former students, meeting new colleagues, and so forth. And the title of my talk has already been announced, so I'm going to go to the first slide. And I think all of you know what structural variation is, right? This is just deletions, insertions, duplications, and translocations, and inversions that change the order or presence or absence of genes and other elements in genomes. And one of the big discoveries, I think it's been underemphasized, from the genome sequencing revolution that has taken place in this century is how much structural variation there is. We had no idea before this. And it's particularly true in plants, of structural variation in plant adaptation, and then plant breeding. The big one I think we've known a lot about, anyone who's tried to cross together of adaptive allelic combinations in nature, and we can use these to, for example, adapt crops to climate change. So I'm going to talk a little bit about some work that one of my former grad students and postdocs, Kai-Chi Huang, did in a sort of a pan-genome analysis in PNAS in 2023. And this is comparing 9 different cultivated reference genomes. And Kai-Chi, by the way, is here, and he'll be talking this afternoon, so if you want a more in-depth look at his study, please go there. And he's now a prof at Sun Yat-sen University. Anyways, this just gives you an idea of numbers. Nothing important here other than to say that if you compare any given cultivated genome with another, this is with oilseed cultivars. Tao Chan will give a plenary tomorrow talking about how different confectionery cultivars are to oilseeds. They're vastly more different. And but you can see that on average, or probably a median would be more correct, there are some large indels, 40,000 copy number variants, a smaller number of translocations, and about resistance genes and so forth have come from log relatives. Now we're seeing more and more genes involved in abiotic stress resistance. But these wild genomes are highly divergent in structure. So this is a comparison of the silverleaf sunflower, which is the sister species to Helianthus annuus, the cultivated sunflower. And it diverged from cultivated sunflower about 1.7 or 1.8 million years ago. And the red arrows just show a couple sort of exemplar. There's an inversion there you can see on chromosome 1, but there are lots of other inversions. There's also translocations, and these are when the sort of lines go between different chromosomes. And so I want to talk a little bit about large translocations. We've known about these in sunflowers for years. And we know in sunflower that wild relatives have been used a lot in breeding. And this is a study that one of my former grad students, Hannah Stempelwolf, he's at the Global Crop Diversity Trust now, and he basically went through the literature and counted up. Selection will do the work for you. And we actually don't see any, as far as I know, large translocations in cultivated sunflower and its progenitor. When we look at other wild species, there's a whole, all of the wild species differ by 6 or more large reciprocal translocations from the cultivated sunflower. This is an example of Helianthus pedularis, which is the prairie sunflower. It's a bit more distantly related. It's been used a lot in sunflower breeding, and it's about 2.5 million years divergence, and you can see that there are 9 chromosomes that actually have these translocations. Of course, you can also see that there's far more inversions, about 60 or so inversions greater than 1 megabase in size. Okay, so what do they do? Well, as I mentioned, we've known this for a long time. They cause reductions in pollen and seed viability. And the big thing is they are independent replicate experiments. You can see that there's very little introgression in the translocation itself. They get removed very, very quickly. So there's a big mystery that I'll talk about another time, but that is how these things become established in the first place. And if anyone wants to talk to me about that, it's something I'm very fascinated with. But I'm going to leave it for today because of time reasons. So I think a much more important, and what I would argue, maybe the most important thing. So the reason that these are established or maintain polymorphic in wild populations is that a couple examples of these. These are, the 2 here on chromosome 1 and 5 are interesting to me because they are segregated high frequency in the oilseed cultivar gene pool. Conditions that can be exploited by breeding. So I thought I would just provide a little bit of evidence about what these things do. So this is from genome-wide association studies of wild species, and this work was mainly done by Natalia Perkovic, a research associate in my lab, and Marco Tedesco, who's a former postdoc, now an assistant professor at the University of British Columbia. And they did what I thought was a Herculean study, which they weren't the only ones involved, but they were the leaders, and did basically a genome-wide association study of 4 wild species, 3 common gardens, 80 plus phenotypic traits, and 1,506 for Helianthus annuus. I thought it was a little bit underwhelming. For example, I sort of lumped different traits together to make it a little easier to understand here, but for example, that inversion 13.1, that's that massive inversion, it mainly affects height, stem diameter, sort of size traits, right? Stem diameter, disc diameter, leaf number. It also affects flowering time. Another inversion, 11.1, affects leaf shape and filary shape. Others, stem color, petiole color. Some affect seed shape, and so on and so forth. Environmental GWAS. And the idea here is environmental GWAS, unlike sort of phenotypic GWAS, uses characteristics of the local environment as the phenotype. And so here again, we're doing environmental GWAS of 4 wild species, 15 soil characteristics that one of my postdocs, Dylan Burge, went around across North America and collected soil samples from the sites where these wild populations occur, and then also 25 climate variables for each site. And the results, I think, turned out to be quite remarkable. What we find is that these inversions are showing strong local adaptation to different environmental characteristics where this occurs. There's one key ecotype that occurs on sand dunes that has these very large seeds. We call it very creatively the dune ecotype. And then the other ecotype we call the non-dune ecotype. And there's gene flow between them. But they accumulate in these inversions, these alleles that provide local adaptation to the dune. This is seed size, flowering time, and cation exchange capacity. You can see the dune-specific traits mapped to the large inversions. And this is, take one of those inversions. Okay, there's a problem in Helianthus annuus, though, with environmental GWAS, and the reason is that environmental variation in Helianthus annuus, because it has this very broad geographic distribution, is correlated with genetic structure, and it was messing up our ability to get significant effects. And so one of my current graduate students, UAU, by the way, for those of you who don't know this, there's a family connection here where UAU is the daughter of our president and organizer, Professor Zhao Jun. And so it's been fun to have her in the lab. And so what she did was she used a different approach for looking at environmental GWAS that essentially accounts for that these things really did have significant effects, that we weren't getting false positives. And I'm going to show some results. Here, this is again, I was just showing the arrow in version 13.01 because you all kind of know that. In this case, you can see that it has a strong effect on temperature differential. So temperature differential is the temperature difference between the coolest month and the warmest month of the year. And so sometimes the trait is called continentality. We always work here. But what you can see is that these things inversions are really doing is almost all of them are providing local adaptation to temperature and precipitation or both. And so these are your climate change inversions. They've over millions of years, these things are millions of years old, they've accumulated multiple, and UA has evidence for this as well, but I don't have time to show, they've accumulated multiple, many alleles within the inversions. They basically pyramided alleles for adaptation to drought or to high temperatures and so forth. When you look to these inversions when you do pre-breeding, well, almost nothing. It turns out that the inversions have very minor impacts on introgression. And so they do introgress into cultivated sunflowers. And the reason is that, one of the reasons is that unlike what we thought inversions caused a reduction in fertility and viability. Well, most of them don't. Most of them have no impact whatsoever that we can detect. And so they are easy to introgress into cultivated sunflower lines, and they don't negatively affect fertility. And again, this was something I was going to talk about a little bit why that is. We don't know. It's one of those mysteries that needs to be figured out. There is a little bit of an effect here. So these, just so you understand these graphs here, these are pre-bred lines that we created about a decade ago. Greg Bowdy, one of my former grad students, created these lines, and then Fernando Hernandez has done these recent analyses in gene content. To give you some idea of scale, there's more variation in gene content between two different oilseed cultivars than there is across vertebrates. Okay? It's just mind-boggling. And so in sunflower, Kai showed that there's about 40-some thousand genes that are found in all of the oilseed cultivars that he, I should point out that this is all a collaboration with NRA and the Sunflower International Consortium of Sunflower Genomics, and then you can see quite a number of genes are dispensable. That is, they vary between different cultivated lines. And these numbers will grow once one of the wild species that has a huge impact. This is in the silverleaf sunflower, Helianthus argyrophilus, and this is a sunflower on the coast of Texas. And what you can see, there's a massive peak at the top of chromosome 6 that affects flowering time, and it's about 30 megabases in length. It's not an inversion. It turns out, if you look more closely, it's a presence-absence variant. And this region includes a really important gene family, the flowering locust T gene family. And this is, and it's the protein of flowering locust T that migrates from leaves to the apical meristem to actually trigger flowering. And in this case, there's an interesting polymorphism for FT1. As you notice there, FT1 is present in the early flowering Helianthus argyrophilus. Work to confirm this, he showed that you can't amplify the FT1 from genomic DNA in the late flowering ecotype, same thing with cDNA, and the early flowering allele actually complements the Arabidopsis mutant, indicating that it's functional. So in this case, it's actually an interesting evolutionary story because sometime in the last 50,000 years, this FT1 gene, I'm not showing this here, actually was acquired from another species. The function, show you just, this is not working. Okay, this slide, there's a compatibility issue. So we're gonna go on. Oh man, this is an interesting slide. It should be, because it turns out that we've run genomic prediction. Yeah, it's hidden in there, some hanger or something. I don't know what happened to it. We found that about 75 to 80% of the time, it was the presence allele that increased the phenotype for a heterotic trait. This would be things like disc diameter or so forth, stem diameter and so forth. And so what this suggests to us is that in about 3/4 of the cases, the missing allele or the absent allele at a presence-absence variant has come from. So ultimately, of course, they come from mutation, but proximally in the cultivated sunflower lines, a lot of that variation actually comes from intergressions from wild species. And so this is from Kai's paper. He's showing that if you look at, you can see particularly in the male lines or the restorer lines where there's more introgressions, you can see many new genes have been introduced through these introgressions. And you also find that in many cases, the introgressions don't have the gene that they should. And it's not because those genes are necessarily missing in the wild species, they're just somewhere else in the genome. So when you introgress the fragment into the cultivar, you end up with a missing gene, and it's those missing genes that seem to be, that we think are the cause of this genetic load that we're seeing associated with the missing allele of presence-absence variants. And some sort of confirmation comes from this in that this is a complicated slide. Basically, it's comparing the effects of introgressions from the primary germplasm, this would be wild Helianthus annulus or exotic material of Helianthus annulus, and introgressions from other wild species. And then we're looking at the effects at 3 different common gardens, and then for a number of traits ranging from plant biomass to number of branches to flowering time to various yield-related traits like head diameter, head weight. Well, I don't want to go too long. And this is just a short discussion about what I talked about this morning. The biggest take-home message I want to leave with you is that these sunflower inversions, which are so incredibly common, they actually represent supergenes that confer adaptation to challenging environmental conditions, including adaptation to drought, heat, and cold stress, as well as low-nutrient soils. And this is not just sunflower. We're finding these things are being found in other crops as well, although I think sunflowers might be the most dramatic. And it should be straightforward to move these supergenes into cultivated lines, permitting rapid local adaptation to extreme climate conditions. There is a downside, though, a caveat, the kryptonite to breeders to Superman, and that is that these inversions are likely to come with unwanted traits that will be extremely difficult to decouple from targeted inversion haplotypes. So let's also talk a little bit about mitigating the impact of reciprocal translocations and gene presence-absence variation. What we talked about today and showed was that structural variants contribute importantly to genetic load and linkage drag, and this is most problematic for introgressions from the secondary gene pool. And so there's a fairly obvious sort of suggestion here, and that is in pre-breeding experiments, the more we focus on the primary gene pool, the better. And so with genomic data, we can actually, there are very few fixed differences between different wild species. You can almost always find an allele that might be frequent in another wild species, a more distantly related one, segregated at low frequency in wild Helianthus androids. So I think you can still find those traits or those genes of interest. Also, I would recommend choosing lines for hybrid production that maximize complementation of introgression in the secondary gene pool, from the secondary gene pool. And this is a fairly easy way to sort of account for that genetic load that's being introduced by these secondary introgressions. And then finally, I would argue, and we're going to see some very interesting talks here in this conference, and I will highlight one by Bing Wang, who's a professor at the Institute of Genetics in Beijing, where there have been some dramatic advances in functional tools that will allow us in the sunflower world to mitigate some of these effects by using new breeding techniques, and then you don't have to worry about linkage driver. And so I want to end by thanking everyone who's been involved with this. I really want to thank the International Consortium for Sunflower Genomics that's been headed up for the past few years by Stefan Munoz. They produced many of the resources that we talked about here today. And then, of course, I've talked about most of the people here that I'm showing in this slide. Much of the funding came from Genome Canada. And I also want to acknowledge my lab. I'm showing sort of an older slide because it shows some of my Chinese students who are here in the audience today. Tai-Chi Huang is in there. You'll recognize him once you see him give his talk, and so is Lashan Gao. So anyways, thank you very much.